What is hybrid dysgenesis? Hybrid dysgenesis is an umbrella term that describes any genetic abnormality or sterility that arise from offspring of parents with different transposable elements. Usually these offspring have a different location in which this jumping gene has inserted itself in compared to their parents. For the researcher's purpose, it's used to describe why the offspring of certain crosses are going to be sterile. And this is really important to keep in mind when you're searching for particular flies from a particular cross if you are a Drosophila melanogaster researcher. Before we look at hybrid dysgenesis, we have to look at practically why P elements are suppressed from mobilization. Let's review how genes are regulated. First of all, genes are transcribed into mRNA, which is then translated into a protein product to express the gene in question. This is a really complicated process that does require some regulation. One way to regulate genes is through suppression of transcription, and this usually will occur in Drosophila eggs. You could have maternal RNA sequences, which are pretty short, that can suppress transcription of certain genes. Also, there can be protein repressors that are found in oocytes or Drosophila eggs that also repress the expression of certain genes. Why would you want to repress the expression of certain genes in a developing zygote? Well, the genes that are usually suppressed or repressed are thought to help control any abnormalities or issues occurring in the embryo's development. Let's look at a small example. So here is an oocyte, and the black line represents just the regular genomic DNA found in an egg cell. We need to repress the expression or the transcription of the gene of interest. So the green sphere represents either a sequence of maternal RNA or a protein repressor that actively will prevent the transcription of that gene so it's not expressed. This similarly functions for P elements. These are naturally suppressed for an egg cell, and it's because P elements can disrupt developmental genes. Transposase is the enzyme that is responsible for moving P elements around, so naturally it is the first target for maternal repression. Here you can see the maternal repressor sitting on transposase's promoter and RNA polymerase not being able to transcribe it. When transposase is not transcribed, it is not expressed, and this P element is not going to move. As a researcher, you may want the P element in the offspring to jump into a different location than where it was present in the parent lines. So you could probably find the maternal repression of transposase annoying in order to accomplish this goal. In order to work around this, researchers use two different lines of P elements, autonomous versus non-autonomous, in the parental lines. Let's first review the difference between the two before we look at which one is going to be in which parent. Autonomous P elements are able to move on their own because they contain the gene for transposase. Once transposase is expressed, it's able to cut out the P element at the inverted repeat sites and help it move to a different location in the genome. Non-autonomous P elements, on the other hand, are not able to move on their own, and this is because they lack the gene for transposase. Without transposase, there's nothing that can cut at the inverted repeats and move it around, so non-autonomous P elements are stuck in place. Now that we have finished our review, let's go over a couple scenarios to see when and where hybrid dysgenesis will occur. In our first scenario, we are going to place an autonomous P element into our female line, and then we're going to put a non-autonomous P element into our male line. So you can see here that in the autonomous P element, there is transposase. So in theory, it should jump around, right? Well, as we know, there is maternal repression of transposase transcription, so this P element is not going to move to a new location. No hybrid dysgenesis occurs in the offspring because that P element hasn't moved. So it's got the p elements in the same places as the mom and dad. We refer to the autonomous p element line as P and the non-autonomous p element line as M. Let's swap it. So in scenario 2, we're going to put the non-autonomous p element into the female line, and we're going to put the autonomous p element into the male line. So it's the one that now has transposase. And if we look at the egg and the sperm cell differences, in the sperm cell, that p element is going to jump because sperm do not have suppression of transposase. So here it's in a new chromosome, which is indicated by green, and you had hybrid dysgenesis occur in the offspring. And that's because transposase was not suppressed. There are some big consequences of hybrid dysgenesis because when p elements move, you have a higher chance of mutations occurring. You have more of a likelihood of chromosomal inversions occurring, which can lead to sterility in these offspring. So I hope you learned a little bit about hybrid dysgenesis and some of the consequences that might occur.